Hello everyone, I am Shurasri Pal. I am a Prime Minister's Research Fellow, and today we are solving this GATE 2018 question from the paper Xl-R, question number 19. And the question states, in garden tea, dwarf plants with terminal flowers are recessive to tall plants with axial flowers. A true breeding tall plant with axial flower was crossed with a true breeding dwarf plant with terminal flowers. The resulting F1 plants were test crossed and the following progeny were obtained. So tall plants with axial flowers 320, dwarf plants with terminal flowers 318, tall plants with terminal flowers 79, and dwarf plants with axial flowers 83. The map distance between the genes for plant height and the flower position is, and we have to calculate the map distance in the unit centimorgan. CM stands for centimorgan. Now let's see how we can solve this particular question. So we know that when there are two genes on the same chromosome, for example here, there are these two genes, capital A and capital B. On the homologous chromosomes, it has two other alleles, small a and small b. And when these two genes are present on the same chromosome, there can be a crossing over in between them, or there can be no crossing over. So if there is no crossing over, we know that these genes will be inherited in its parental type only. So if there is no crossing over, only the parental type of gametes will be formed. So here we can see that what are the parental types of gametes? When there is capital A and capital B both on the same chromosome, and small a and small b on its homologous chromosome. So there is no exchange of the um, chromosome parts, which is why the dominant genes remain on the same chromosome and the recessive alleles also remain on the same chromosome. So there is no crossing over in between them or no recombination. However, what is crossing over? Crossing over is the exchange of the chromosome segments, and crossing over is the basis for recombination and also for linkage. So here we can see how this crossing over forms, and this is something which we call the chiasma formation. When there is exchange of the or there is overlap of the non-cystic chromatids of uh, two homologous chromosomes. So these two are the homologous chromosomes, and there is an overlap of the non-cystic chromatids that we can see here, and this forms a X kind of a structure, a chiasma kind of a structure, and there is an overlap. So what happens because of that? These in these chromatids, we can see there is an exchange that has happened, and because of that, capital A has paired with small b, and small a has paired with capital B. So these two types, these are the recombinant types, whereas when there is capital A and capital B together, that is the non-recombinant type or the parental type, and when there is small a and small b together, that is also another non-recombinant or the parental type. Whereas these two, the capital A and small b, that is a recombinant chromosome, and small a and capital B, that is also recombinant chromosome. So that is crossing over. So this is, I will again mention this, that when there is crossing over, there is meiosis with crossing over, then there are four types of gametes that will be formed. So here we have seen that there are only two types of gametes that have formed, two types of parental gametes that have formed, and they resemble the parental type. Whereas when there is crossing over, we can see there are four types of gametes that will be formed, and two of them will be the parental type, and two of them will be the recombinant type. So these two are the recombinant types that you can see here that forms due to crossing over. Now, when we are discussing crossing over, if the genes, if the two genes on the same chromosome, they are located close to each other, then does crossing over happen between them? Or does crossing over happen between all the genes? So that question was actually addressed by Morgan, and he observed that it's not true that crossing over occurs between all of the genes. So there is also a phenomenon which is called linkage, or there are linked genes. There are linked genes. So capital A and capital B, they are if they are linked, if these two genes they are linked, then what will happen? Then only the parental types of gametes will be formed. Capital A, capital B, and small b. These are the gametes that will be only formed if there is linkage. That means if there is full linkage and if there is no crossing over between the two alleles or the two genes on the same chromosome. So only parental types of gametes will be formed. But if there is linkage with recombination, so which means that there is a percentage of linkage between the genes, but um, there is also crossing over that also take place in between them. So that is linkage with recombination. So if there is linkage with recombination, then these two alleles, there will be the parental types of alleles will also be formed, and the recombinant types of alleles will also be formed. So we can see here that this one and this one, they are the parental types of gamete. Parental type, and this one is also a parental type of gamete. So capital A, capital B, and small b, they are the two parental types of gametes, and these one are the recombinant types of gametes or the crossover types of gametes. Capital A, small b, and small a, capital B. That is also a recombinant type of gamete. Now, here we will obviously see that since the genes are linked, so there is a percentage of linkage in between them. So that is why these gametes will not be formed in equal ratio. So if there was crossing over and if there was no linkage, so if there was no linkage, then we will see the gametes in the ratio of 1:1:1:1. So which means the recombinant type and the parental type will be formed in equal ratio. But since there is linkage, if there is linkage, then the parental type will be produced much more in number than the recombinant type, because the genes are linked and the chances of recombination are low. Okay, so which is why the parental type will be produced in much more number than the recombinant ones. So this is something that we will need in the in solving the questions related to the non-Mendelian genetics. So the parental types will be produced in much more number than the recombinant types. This you must remember always. Now coming to the question, now let's get back to the question. Okay, so the true breeding tall plant was crossed with a true breeding dwarf plant. The true breeding tall plant with axial flowers was crossed with a dwarf plant with terminal flowers. And it is also mentioned here that the tall plants and axial flowers, they are the dominant characters. Tall and axial, they are the dominant characters. So what we do is here we assign tall with capital T. So the dominant characters we usually assign with capital letters. So tall we assign with capital T, and axial we assign with capital A. Okay, so tall with capital T, axial with capital A. And dwarf plants we assign by small t and terminal flowers by small a. So tall plant and axial flowers are dominant over dwarf plants and terminal flowers. So what we do is in the parental generation, we take a true breeding tall plant with axial flowers. So we will write it here. True breeding tall plant with axial flowers and cross it with true breeding dwarf plant with terminal flowers. Okay. So true breeding means they are the homozygous, and that is the parental generation. So in the F1 generation, then what happens? The question reads that the resulting F1 plants were test cross. Okay, so first of all, what will be the F1 genotype? In the F1 generation, we will get all same types of plants with the genotype capital T small t, capital A small a. Okay, so this is the F1 genotype, and all of the plants will be tall plants with axial flowers. Okay, so that will be the genotype tall plant and axial flower because they are the dominant ones, the dominant phenotypes, they will be expressed. Now, these F1 plants were test crossed. What does it mean? Test cross means that crossing the particular plant with with recessive, homozygous recessive plant. So, what is homozygous recessive here? The recessive characters are your dwarf and terminal flowers, right? So, therefore, uh, this is the recessive plant with which we cross the F1 plant. So, that is the test cross. Now, what are the combinations that we can expect here? What are the phenotypes that we can um, expect here? So, if we draw, so first of all, what will be the gametes from this F1 plant? So the gametes will be since um, we we will consider independent assortment here. Uh, we will there will be formation of four types of gametes: capital T, capital A, small t, capital A, capital T, small a, and small t, small a. So these will be the four types of gametes that will be produced from the F1 plant. And what will be the gamete? What will be the type of gamete that will be produced from the recessive homozygous recessive plant? That will be only small t, small a. That will be the gamete from the homozygous recessive plant because there are no other alleles. So the gamete from the homozygous recessive plant will be small t, small a. Now, what are the genotypes? Therefore, in the F2 generation, what will be the genotypes? Capital T, small t, capital A, small t, capital A, capital T, small a, small t, small a, small a, right? So these are the genotypes, and let's see the phenotypes also. Let's write the phenotypes also. It will be tall and axial. It will be dwarf and axial. This one will be tall and terminal, and the last one will be both recessive, dwarf and terminal. Now, if there was independent assortment and there was no linkage, that means if there was crossing over, if there was 100% crossing over, then we would expect a ratio which is 1 is to 1 is to 1 is to 1. That will be when there is no linkage. So that is the um, test cross ratio of the dihybrid cross of Mendel. So what Mendel was also observed, Mendel had also observed that if there is no linkage between the two genes on the same chromosome, the test cross ratio of the dihybrid cross will be 1 is to 1 is to 1 is to 1. But if there is linkage, then this ratio will not be maintained. Because why? Because as I mentioned here, that if there is linkage, the parental types will be formed much more in number than the recombinant types. So the parental, what are the parental types here? Let's denote them. This one is the parental type, the tall and axial. This one is one. This is one parental, and the other parent is this one. That is the homozygous recessive. So that this one is the parental. And these two are the recombinant types. And if there is linkage, then the parental types will be produced much more in number than the recombinant types. Now let's see what the question says. If you go back to the question says that tall plants with axial flowers will be 320 in number, dwarf plants with terminal flowers 318 in number, tall with terminal flowers 79, and dwarf with axial flowers 83. So here we can clearly see that it does not maintain this 1 is to 1 is to 1 is to 1 ratio. If there was 1 is to 1 is to 1 is to 1 ratio, all will be equal in number, right? But here this ratio is not maintained. And we can see that these two groups, that the the tall plants with tall plants with axial flowers and dwarf plants with terminal flowers, they are maximum in number. So we can easily assign them as the parental group. The tall plants with axial flowers and the dwarf plants with terminal flowers. Why am I saying this? You know here that these are the two parental groups, the tall plants with axial flowers and the dwarf plants with terminal flowers. But in some of the cases, in some of the problems, you can see that you do not know what is the parental type. Then this ratio will actually help you to understand, these numbers will help you to understand which are the parental types and which are the recombinant types. So the large numbers, the majority numbers will be always the parental types. And the other two will obviously be the recombinant type. And that is something, the same thing we observed here also. That is the tall and axial, that is the parental type, and the dwarf and terminal, they are also the parental type. Okay, so I'm writing it in the next page. The tall and axial, that is 320 in number, that is one parental type, and the dwarf with the terminal class, that is also a parental type, that is produced with number 318. Okay, so these are the two parental types. Now coming to the two recombinant types, which are tall and terminal, that is produced in 79, and the other recombinant type is dwarf and the axial, that is 83 in number, right? So the ideal ratio of 1 is to 1 is to 1 is to 1 is not maintained. That is the ratio when there is no linkage, that is not maintained. So therefore, obviously, there is linkage here. Now, what do you have to calculate? The question is asking to calculate the map distance between the genes. Now, for calculating map distance, the first thing that we have to calculate is the recombination frequency. So, we are continuously saying that these two are the recombinant types, right? These two are the recombinant types. So, how do we calculate the recombination frequency? The recombination frequency will obviously be the sum total of the number of recombinants divided by the total number of progenies. Okay. So the recombination frequency will be the total number of recombinants divided by total number of progenies. So how do we calculate that? The total number of recombinants is 79 plus 83 divided by total number of progenies is 320 plus 318 plus 79 plus 83. And if we calculate that, we would get 162 divided by total number of progenies, that is 800. And the calculation will be 0.2025. Okay, so that is our recombination frequency. What is our recombination percentage? The recombination percentage will be 0.2025 into 100, that is 20.25%. That is the percentage of recombination. And the percentage of recombination is actually the same unit as the map distance. So basically, 1% of recombination is equal to 1 map unit distance between the genes. 1% of recombination is considered as the 1 map unit distance, or it is also considered as 1 centimorgan. So this unit is centimorgan. It is in the name of the scientist Morgan, who first worked with linkage, right? So 1% of recombination is known as 1 centimorgan. This is something that you must remember for these type of sums, that 1% of recombination, 1% of recombination means 1 centimorgan unit. Now the map distance here in this particular question is asked in the unit centimorgan. Okay, the map distance between the genes, plant height and flower position. So the recombination percentage between these genes, the plant height and flower position we have calculated, and which we get as 20.25%. So therefore, what will be the map distance? The map distance will also be the same, because 20.25% means 20.25 centimorgan. So our answer is 20.25. Okay, so this is our answer, 20.25 centimorgan. Okay, for more information, you can visit this particular in detail course. The lectures are Mendelian and non-Mendelian genetics. Thank you so much.